Hey, good morning. Um, so today we are going to continue talking about the process of gene expression, starting with the transcription of DNA into RNA. And we're going to talk about some of the different types of RNA molecules that are made, and importantly, take a look at why our genetic sequence is arranged the way it is. The basic requirements for being able to code things like amino acid sequences that make proteins based upon the original DNA sequence that's in our genes contained in our chromosomes. So to start off with, I wanted to just review with you where we are in the semester. I should point out, I think the PDF version of today's lecture that was posted that I had given to your TIs was outdated because I think it still had the schedule from last fall in it. If that's true, I'm going to give the TIs the updated version of today's lecture, and then they will go ahead and swap that out on Laulima so that you can then download the updated version. I mean, most of the content of the lecture is the same, but this slide I think might be outdated. Anyhow, so let's take a look at this. So here we are at the start of week 4. So we are going to talk about RNA processing and the process of how ribosomes are involved in translation. We're going to continue talking about that through the week, about how the products of translation are impacted by things like mutations in our DNA, and how the proteins that are made by ribosomes through the process of translation then get further processed and turned into mature proteins. So this leads us into next week, which will be our first exam. Right, so the way the exam is going to work is it's divided into 2 parts. On Wednesday during class time will be the multiple choice part. That's all done on Laulima. Then later next week, either on Thursday or Friday, depending upon when your recitation section is, you will then do the free response written part with the benefit of your TI being there to help proctor you during that session. So we will talk in more detail about this this week. Okay, I don't want to spend a lot of time today talking about it because it just means I'm going to have to repeat myself later on. But your TIs will also discuss the exam format with you this week. All right, so this is just to give you a heads up that our first exam is coming up, so you can plan accordingly. Okay, so we had been talking about the fact that our DNA sequence and our chromosomes is representative of different chunks of information. These different chunks of information are our genes, right? Our genes that are separate sets of instructions to make something. I had described, for example, one way of thinking about it as genes being like the cookie recipes in a cookie recipe book, right? So each recipe is its own discrete set of information to make a particular cookie product. We can think of the cookie product, the finished product, as being the finished protein, for example, that then may have various functions inside of the cell. So we said that the way that our cells utilize this information is not by taking the original cookie recipe, the original DNA, and directly making proteins from that, right? Because there are certain disadvantages to that. For example, you want to protect your original DNA, because if the original DNA got damaged, right, then you're in big trouble if this information somehow got compromised. Also, because there's only one chocolate chip cookie recipe in this book, if you really needed to make a lot of this particular product and all you had was this one physical copy of the original recipe, that might slow you down because you would not be able to share it with other friends who all have their own kitchens and would be happy to help out, right? If you were stuck with this, just this one. And also the fact is, right, that the information contained here is static. It's not changing. Right, the instructions on this piece of paper will never change just as long as the book is protected. So that's why we said that we make an intermediate molecule, and that is RNA. And we said RNA is a photocopy, a transcript of the original DNA that gives you all kinds of flexibility, right? You can make many copies, many RNA molecules from this, and now be able to make many batches of cookies or a lot of protein at the same time. If one of these gets damaged, it's not a big deal because you have many other photocopies of it. And importantly, you can edit this, right? Or your friends could edit this, cross out certain things, make substitutions, and make slightly different variations of the finished cookie. So in this illustration here on the left, we had looked at this before. We said that our chromosomes are our single strands of DNA, right? We have different chromosomes. So even if you looked at chromosome 1, that is just one long piece of DNA, but it's not naked DNA. We've already learned it's packaged up as chromatin that allows it to be condensed so that your whole genome can fit in a teeny tiny cell. But importantly, by wrapping it up, we also provide a way for the cell to control what regions of the chromosome, right, what stretches of genetic sequences or genes are available at a given time in order to be transcribed and made into the protein product that they code for. So in this illustration here, when the chromosome is kind of wrapped up tight, we say that that's heterochromatin. So if you look at the bottom here, when the chromatin is tightly packed up, it's heterochromatin. And as you can imagine, that makes it very difficult for the genetic information in that particular region of the chromosome to be read. When it is unraveled, certain stretches of it, like, for example, if this ball of yarn represents heterochromatin, certain parts of it have been teased out, right, and are now available to be read. Those are euchromatin regions. So at any given time inside of our cell's nucleus, our chromosomes are in a dynamic state of different parts of different chromosomes being heterochromatin, different parts being euchromatin, right? Either tightly packed or unraveled. Last time we went through this kind of quickly, so I wanted to spend a little bit more time on this today because this is some important concepts here. We said that along the chromosome, if you looked at it, you cannot tell what is a gene or what's not a gene. Right? When we look at information like a cookie recipe, you know, our brains are very good at interpreting what we see, right? We can tell that this is one cookie recipe because there's certain features on it that visually help us identify, yeah, this is meant to be one set of information that's different than what was on the previous page and what's different than the next cookie recipe on the next page, right? Now, how do we do this? Obviously, it's because we're used to reading. Right? So we're able to recognize that you're supposed to read from one direction, usually top to bottom here, from left to right, because it's English, right? Of course, in other languages, it might be reversed. Right? So we do that because we interpret it. But how would something like a stretch of DNA, right? That's just a whole bunch of A's, T's, C's, and G's, right? What method do we have for your cell to actually realize where a discrete set of information starts and ends? And we said the way it does this is by certain regions containing sequences that are very specific and that bind certain proteins. So certain proteins are the right shape to bind to these particular sequences, and the eventual collection of different proteins around those special sequences will start the process of transcription. So in this illustration here, we said that these regions in the dotted line are what we call the promoters of a gene. This would be that sort of the header, right? That lets your cell know that this is the start of a piece of information that should be transcribed. And these big gold blobs here represent RNA polymerase complexes that will eventually end up sitting on these promoters, and they begin transcribing, right? They are the photocopiers making the photocopies. But keep in mind, right, even though this photocopy is identical in information to the original DNA, right? This is in a different molecular form. This is RNA, not DNA, right? So as the RNA Pol II is reading the template strand, you can see it is making an RNA strand based on the template strand, right? Right. So the DNA then is transcribed or photocopied by RNA polymerases to make different types of RNA. Right? And the three main types our messenger RNA. So this would be the full sort of cookie recipe itself, right? That gets eventually translated into the finished protein. But the two other important types of RNA that are also transcribed from DNA at different particular spots in the genome, right? There are genes that code for tRNA and genes that code for rRNA, transfer RNAs and ribosomal RNAs. Right? And the function of the tRNAs and rRNAs are to work together to help with the process of translation so that you can make finished proteins from the mRNA message. So how these different special promoter regions look, again, we kind of blew through this kind of quickly last time, right? Each one represents a highly conserved sequence in the DNA. So for example, probably the most obvious one is a TATA box, which if you were to look at the DNA, it'd be a stretch of TATAAA. That's very close to maybe 31 base pairs upstream of the actual start of transcription. So this line going across here is the DNA, the template DNA that's going to be read. Right? This initiator site, you can see, has this type of a sequence in it, right? And that helps position the RNA polymerase II over it. And you have these other sites here. So there are 4 basic elements that the majority of promoters share. There's this TF2B recognition element, which is also called the BRE. So it looks kind of like this. You either would find a GGG or a CCA at this site, followed by a CGCC, right? And that would be really close to this TATA box. Each one of these colored boxes, because of its DNA sequence, attracts a particular protein to bind there. And these proteins that bind to the DNA directly and help transcription, we call transcription factors, or TFs. Right? So that's what this little cartoon up here is meant to show, that because this DNA is in a stretch of euchromatin, that means it's kind of been unwound from the nucleosomes and the DNA strand is exposed, then these different proteins that are floating by can match up because they are the right shape to bind to something like the TATA sequence or the BRE sequence, and they pile up, right? They pile up in a very particular coordinated way that's spaced apart by where these different DNA sequences are at the promoter, and eventually they recruit the big RNA Pol II complex, right? Because as they're piling up, they form the right three-dimensional shape for the big RNA Pol II, polymerase complex to also sit there, and it puts the RNA Pol II at just the right spot so that the RNA Pol II starts transcribing, right? It starts reading the DNA and it starts making this RNA strand coming off of it. Okay, right, so that's what you see here. So once these transcription factors have piled up, the big purple RNA Pol II complex gets attracted and it sits there, and the RNA Pol II starts going down this length of DNA and starts transcribing. Right. So I know this concept of transcription factors binding to DNA might seem kind of weird because you think to yourself, well, I don't get it because the DNA is just like this double alpha helix, right? And you saw that there were sort of major grooves and minor grooves because it's not a perfectly symmetrical, but you never really think about the fact that there's actually a very specific little landscape of shapes that are determined by the actual base pairs that are involved in that stretch, right? Very teeny tiny fluctuations in three-dimensional shape. And that's what allows you something like this. This is a kind of a cool, puffy, I don't know what this is, plush model. I don't know where you get this from. I'd like to have one because I think it's kind of nice. But it shows you each one of these different colored blobs is a different transcription factor protein, right? Remember, each one of these is a chain of amino acids. It's a polypeptide chain that is folded up in a particular way so that this green blob, whatever this is, let's pretend, for example, this green blob is the thing that's meant to bind to the BRE sequence, right? So in the DNA here, in this green stretch, you would find GGG, CG, CC, and you can see this green blob, this transcription factor, is just the right shape so that it'll fit in there because of those base pairs going across, right? So very subtle little differences in three-dimensional shape. And that BRE might be kind of just upstream of, let's say, the Tata box or something like that, right? So maybe that's where this protein will bind. And you can see that as they begin to pile up, because they're attaching to the particular short little transcription factor binding sites that they recognize, eventually the shape of this thing is able to recruit the RNA Pol II, which might be the big gray thing in the back here, right? So the RNA Pol II normally would just float by, but if you have the right pile of proteins forming here, the RNA Pol II gets put in exactly the right spot, and the RNA Pol II then begins to sort of unwind the double helix and read the template strand. Right? Read the template strand, and you can imagine the big gray blob here would be maybe moving off to the right of the screen, and you'd have an RNA strand coming out of it based upon the sequence of the template strand in the DNA that it's reading. And the RNA strand would be your photocopy, your transcript, right? It's the first transcript that's made from this DNA, so it's our primary RNA transcript. All right, so this brings us back. Let's step back a moment here, right? And try to kind of put these pieces together. How does this all fit in? Remember, this is what we traditionally call the central dogma or central paradigm of molecular cell biology, which just has to do with how we think about information flowing. Right? Information flows from DNA through an RNA intermediate, right? And eventually being converted into proteins. Right? And so in this figure here, we have two of the four main families of macromolecules, right? Nucleic acids, such as DNA and RNA, and proteins, like the long chains of amino acids that make a protein. Remember, the other two main families of macromolecules that we've been studying as being important for our living cells are what? The sugars and carbohydrates, right? And the lipids and fats. So you say, how does that fit in here, right? So our DNA does not code for how to make sugars necessarily. So our DNA doesn't have the code to make like a little glucose molecule, right? Our DNA also doesn't have the code that gets somehow converted into a fatty acid or a lipid, right? But yet those are vital to our cells. How do they fit into this picture? Well, sugars and carbohydrates we get from the outside, right? We eat things to get sugars and carbohydrates, and we eat things to get fats, right? Fatty acids, things like that. So that's how they get into our body. And once they're in our body, they are manipulated. They are worked on by what? They are worked on by proteins, right? So proteins, when they are made, many proteins function as structural elements, like the keratin protein that makes your tough skin, right? That's a structural type of protein. Many proteins are meant to be tiny little proteins that float around and pass signals from cell to cells, tissue to tissue in your body, right? But other proteins function as little machines that can do stuff. For example, we've already looked at a couple of examples of these. Those big polymerase complexes that copy DNA or copy DNA to make perfect copies of DNA for DNA replication, or what we've been talking about here is RNA polymerases that can read DNA and make RNA, right? Those are complexes of proteins that are working as machines. So think about it in this way, right? The four big groups of macromolecules that we study, so nucleic acids, proteins, sugars and carbohydrates, and lipids and fats, right? All four are vital for our cells to function. But really, the information that's encoded for in our DNA is just coding for things like RNA and RNA that might be an intermediate to be made into proteins, right? So that's why protein biology is so important, because it's the proteins that will then go off and do things with the sugars, with the fats, in order to make everything work. Right? So we also said that messenger RNA is the full cookie recipe that needs to be translated into the finished cookie, right? So that contains the information for the actual amino acid sequence that's going to fold up and make a protein. But other RNAs like tRNA and rRNA, right, they function just as small RNAs because they are parts of machinery such as the ribosome that we will look at later today, that is vital for turning a messenger RNA into a finished protein, right? Because the ribosome, which is again another big complex of proteins mixed with these special rRNAs called ribosomal RNAs. So that's why you see the ribosome is shown here. This big blue region is one big blob of protein, and the yellow region is another blob of protein, actually several protein subunits. And the little strands here are the ribosomal RNAs that help keep this ribosome in the right shape so that it can read the messenger RNA and translate that into your finished protein. Right. So in other words, these would be like your friends who you give the photocopied cookie recipe to, and then it's up to them to actually go to their kitchen and make the finished cookies. Right. All right. So let's look at the ribosome here. So the ribosome is this massive complex made up of protein subunits and this special type of rRNA called rRNA. Ribosomal RNA. In this cool rotating figure here, which, you know, I just got this off of Wikipedia, you know, but it's a nice animation, so I use it. The blue things are the proteins that make up the ribosome. The brown squiggly things are the ribosomal RNAs, and you can see how they work very closely together shape-wise to create this big, massive complex. And again, the amazing thing about this, right, is this big ribosomal complex has to be this exact shape in order to work, because this is the machine, right? And the different parts of the machine have to be in the right position in order for it to carry out its function. The little green dot, which as it rotates around, you might catch a glimpse of it hidden in the middle here. There you go, right? That's the actual site where the messenger RNA is going to be fed into, and then out of the top of this thing, you're going to have a chain of amino acids that are growing longer and longer, amino acids that are being linked together based upon the messenger RNA that this big ribosomal factory is working on. So let's take a look at the rRNAs that make up the ribosome. So there are three main types of rRNA pieces. They are the 18S, the 5.8S, and the 28S. Okay. So these are distinct rRNA pieces that are all kind of coming together to help make this ribosome. They are the squiggly brown things in there. So rRNAs are transcribed from DNA. They're transcribed from very special regions of different chromosomes. I think 5 or 6 of our chromosomes have these rRNA regions, or the DNA sequence for these rRNA regions contained on them, like chromosome 13, 14, I think maybe 21. But these are a little bit different than your typical gene that codes for, let's say, a protein, because these regions look like this. They're highly repetitive. So what you see here, you can see a repeating pattern along this chromosome. You know, maybe this is part of chromosome, I think 13 or 15 or something like that, where you code for these rRNAs. If you were to look at just one of these repeating patterns, it would look something like this, right? So this is one segment of DNA that's repeating over and over again with sort of a little stretch of a spacer DNA in between them, right? So the blue parts contain the information for the rRNA. And you can see part of this is the information for making an 18S, a 5.8S, and a 28S. And they're all on the same piece of DNA. So when this gets transcribed by RNA polymerase, what do you get, right? You get this one pre-RNA transcript, right? The original photocopy that comes off of this that contains, right? So this is one stretch of RNA now, one stretch of RNA, right, that has these different rRNA pieces in it. This then gets processed This one piece of RNA gets cut up, and the 18S, 5, 8S, and 28S pieces of our RNA are basically punched out of it or cut out of it. So why would our cells do it this way, right? Clearly, when you look at the way this is set up and the fact that there are these many, many tandem repeated regions, right? Clearly, there's a lot of focus on making a lot of these small little rRNA pieces at the same time. Right? Now, hopefully you can understand that that's because these small rRNA pieces are a crucial part of making the ribosomes that are in charge of taking messenger RNA and making a final protein from them, right? Your cell has to be making proteins all the time. Otherwise, your cell dies, right? Because so many of the critical functions of your cell is dependent upon proteins for structure, for signaling, and to work as little machines to handle the other types of molecules inside of your cell. So we make a lot of rRNAs at the same time. That's why they're arranged in these tandem repeats. And also, that's why that you have 3 pieces of rRNA made from the same transcribed copy. Why? This way, you're making the same number of each of these pieces at the same time, right? So that's the way your cell controls that you have plenty of these different small rRNAs to make different ribosomes. And it's a good way to control their production because you wouldn't need, for example, a ton more 28S's and very little 18S's. No, that wouldn't make sense. You need to have them in the appropriate amounts so that you can make these ribosomes. Think about it this way. It's kind of like when you buy like a brand new, I don't know, like a board game from the toy store, right? And maybe some of the playing pieces are little cardboard, little playing pieces. You know, they oftentimes come on one piece of cardboard with the little pieces printed on them with little perforations, right? And then you just punch them out. Right? The benefit of the manufacturer making it that way is it's a much cheaper, efficient way for the manufacturer, the toy factory, right, to make these so that it's just up to you, the customer, to like punch them all out. So that's the same thing with these ribosomal RNAs. Evolutionarily, the benefit is it's a very efficient way to make all these little pieces if you just make all 3 at the same time from one of these transcripts, and then you essentially punch them out and your cell can use them. Okay. So what we see here is wonderful evidence of just how much of these rRNAs are made. Here is a strand of DNA. So the big sort of dotted line that goes all the way through here, there's one here, and there's one that kind of comes up over here. That's the DNA. This is a region of these rRNA coding sites. Right? On it, you can make out all these little black dots. These little black dots are different complexes of proteins that are working on this region here. And you can see it's pointing out to you that one of these dots here is an RNA polymerase complex that's going to read down the DNA and make these primary transcripts in the form of rRNA that are then going to be further processed to have the different pieces of the rRNA punched out. So the RNA polymerase starts here, and in this figure, it would be moving down from left to right. Right? And as it moves down from left to right, you can see this is not just one RNA polymerase that's like somehow captured time-lapse wise, because maybe you didn't think about this, but there's nothing to stop a whole bunch of RNA polymerase complexes all working on one gene at the same time, getting down at the promoter region, right? And when one complex starts moving down, transcribing, the transcription factors that are building up at the promoter region, right, can recruit another RNA Pol2 complex and start moving down, and another one, a whole row of them lined up, moving down the DNA, each one in charge of making its own copy from the DNA. And that's what you see here, because as they move down, there's a whole string of RNA polymerase complexes moving down the same segment of DNA. This squiggly thing coming out of them is the primary transcript in the form of rRNA. That's why the ones that are still over here, like the RNA polymerase as it starts, the long tail of rRNA coming off of it would be pretty short, right? But as they move down, right, right, the rRNA coming out of them gets longer and longer. You can see here on this cartoon, picture it this way. This green thing is the RNA polymerase moving down the strand of DNA, right? As the DNA gets into it, it unwinds, and the RNA polymerase is able to read the template strand of the DNA, and it assembles, right, nucleotides, RNA nucleotides, and the growing chain of the RNA is going to get longer and longer and longer the further or farther down this DNA the RNA polymerase gets. So by the time you get to the end over here, right, you have one complete primary transcript now that has been made, right? So I think this illustration is really cool because it means you can actually see the process of transcription happening. And it's a nice reminder, right, that this is a very dynamic process involving oftentimes many transcripts being made from the same coding region of DNA. Right. So let's shift over to tRNAs. Right? So tRNAs are also, of course, read from different parts of our genome where the DNA codes for, right, the right sequence to make these tRNA molecules. Remember, tRNAs, rRNAs, they still have to be made from DNA sequence. So in this case, the primary transcript of tRNA looks like this. Right? So this is now strung together a whole bunch of RNA nucleotides, right? The sequence was determined by the DNA that codes for this. And you can see RNA can form some secondary structures just by itself. Because of course, RNA, you have bases that are across from each other. So C's can still pair with G's. A's would be pairing with uracil, right? Because in RNA, you don't have the thymines, you have uracils instead. So you start getting these little hydrogen bonding features that begin to shape the tRNA into this kind of cloverleaf kind of shape. Right? This tRNA has to be edited further, right? Because remember, the benefit of RNA is it can be edited further, and the finished tRNA molecule looks like this. So it really does look like a cloverleaf. And has the shape because of the hydrogen bonds. Right? So again, think about it, right? It would not be this shape if it were not for the fact that the nucleotide sequence in it has this G across from this C and let's say this A across from this U, right? So that they line up and help form this shape here. Okay. We're going to study the tRNA structure in more depth in a later lecture, but today I'm just introducing the rRNAs and tRNAs. All right. So we said that rRNAs and tRNAs are going to work together to help your cell take the messenger RNA or mRNA message, right, which is the actual recipe for the finished protein, and somehow translate that, in the form of this factory, this ribosome, right, that's going to read the mRNA and put together a chain of amino acids. So that chain of amino acids, of course, itself is encoded for by the original DNA sequence. And so how is it that our DNA sequence of A's, T's, C's, and G's, right, just different combinations of those four nucleotide bases, can contain all the information you need to actually specify what amino acids should be strung together to form a protein? Let's back up and review something about protein structure. Right? We said that many proteins in our body function as three-dimensional shapes because they have a tertiary structure, right? A tertiary structure that can then maybe assemble with other protein tertiary structures to make a quaternary structure. So those big RNA polymerase complexes or DNA polymerase complexes are quaternary structures of different protein subunits, right? Likewise, the ribosome itself that we're going to talk about in detail is a big quaternary structure of different protein subunits mixed in with ribosomal RNAs that help make it its right shape. But remember, if we go backwards a step, the amino acid sequence is the primary structure. So we have 20 different amino acids that are used in our bodies, right? Life on Earth evolved on Earth to use 20 amino acids. And those are the ones that you've been studying, the different R group side chains. So how do we specify 20 amino acids using just 4 DNA bases? So obviously it's some combination of bases need to code for a particular amino acid to be put in that next spot of the growing peptide chain. If we look at the DNA, remember the DNA is double-stranded here. You have the 2 strands that are running anti-parallel to each other. Recall that when the RNA polymerase is going to read this, it's going to unravel this region and it will read the template strand, right? So RNA polymerase reads the template strand of DNA, right? And that's how the RNA polymerase knows what nucleotide, what RNA nucleotide to put next in the growing RNA strand that's coming out. But for convenience sake, we often refer to the coding strand, right? Because the coding strand is more intuitively like the same letters as the RNA. Right? So let's see here. So this mRNA has to code for different amino acids to be strung together. What's the bare minimum number of A, T, C's, and G's that you would need to use in order to have enough information to specify 1 out of 20 amino acids to be in the next position, right? Well, let's see. Could you get away with using 2? What if every 2 bases was meant to code for an amino acid, right? So in this first position here, that happens to be an A, you can have 1 of 4 choices, right? In the second position, that's a uracil, it could be 1 of 4 choices, right? So that would be 4 times 4. No, that's not enough, because 4 times 4 means you could only code for 16 different possible amino acids, right? And we know that no, we need to code for 20, right? So every 2 bases doesn't give you enough choices. What if you say we code with 3? So every 3 nucleic acid or nucleotides here, right? That would be 4 times 4 times 4. That would give you 64 possible different variations. So that's way more than we need. So that's fine, right? Because if 2 is not enough and 3 is too much, well, life just evolved to use 3, right? And so that also implies that you have a lot of redundancy, meaning you're going to have a lot of extra combinations of 3 nucleotides or 3 bases that can also stand for the same amino acid, right? So every 3 bases gives you more than, that's why life evolved with a triplet code, right? So in our DNA, every 3 nucleotides, right, when it gets transcribed into every 3 nucleotides of mRNA, that is enough information for your cell to know that at that spot in the growing polypeptide chain of the translated protein, a particular amino acid out of the 20 needs to go there. So here's the genetic code. I think you guys have all seen this before, right? So this kind of redundancy of having way more possible triplet combinations, you know, we say that it's a degenerate code, meaning that there's redundancy in there. So there are 61 codons out of the 64 that code for amino acids. And then you have these special codons, right? So every 3 of these is called a codon that codes for different functions. For example, AUG codes for methionine, but that's also the start of translation. Right? So it's at the AUG in the mRNA that translation is going to start. And that's where the polypeptide chain of amino acids is going to start being put together. Likewise, you have these 3 stop codons, UAA, UAG, and UGA. So in other words, when the mRNA is being read through the ribosome, when the ribosome reaches one of these stop codons, that's where it knows to stop putting together amino acids. And whatever chain it has put together up until then, that chain of amino acids, that polypeptide chain is the basis, your primary sequence of your protein that is then going to fold up and maybe make a 3-dimensional shape. So you don't need to memorize this chart. So on the exam, you know, you'll be given this table. I mean, I don't even, you know, I don't memorize this because it's kind of silly, because if you need to translate something, you just look it up, right? But this is how it works, right? So if you look at the DNA here and the mRNA, you see there's an AUG. So that is methionine. So the way you look it up is on the table here. The first position that you want, right, is either going to be a UCGA, right? So you look it up along this side first, right? The first position. In this case, if we're looking up AUG, you're like, oh, okay, it's an A. The second position up here is a U, and the third position on the right here is the G, right? And you can see AUG is right here, and that codes for methionine. All right, so if you looked at the next one over here, GGC, right, you'd start from the left, G, the next position is a G, so you'd be over here, and then C, right, you'd be over here. All four of these variations of the codon, this is the whole redundancy thing, code for glycine, right? So then you, oops, sorry. Oh, my animation doesn't work. So this should be a glycine in here, okay, etc. Okay. So be familiar with how to read this table. I mean, look, it's not that hard, right? But if you don't practice it and you don't know how to do it in an exam, it's kind of a shame because these are like easy points, just knowing how to read this table. Right. So when the mRNA, or when the first original primary transcript of RNA is being made here by the RNA polymerase, right? So now you have this molecule of our RNA. It has to be further worked on, right? In other words, this is happening in the nucleus of your cell. Your ribosomes that eventually translate the mRNA, they do that in the cytoplasm. So how does this rRNA fragment, make it from the nucleus out to the cytoplasm? It's not going to just float around. Your cell doesn't just hope that things are floating around and eventually they get to where they want to be, right? Because life wouldn't work like that. The RNA has to be prepared. It has to be processed, and it has to actually be sent out of the nucleus. One of the first steps that happens is that at the 5' end of the RNA transcript, so in this case, these are the last two nucleotides at the end, at the 5' end of this RNA transcript, okay? So you can see there's two nucleotides here. They get methylated, right? So these methyl groups get stuck onto the C2 position. So you can tell this is RNA, right? Because in DNA, this oxygen wouldn't be here anymore. Right? So this allows it to be methylated. And at the very top, they get a backwards methylated guanosine nucleotide stuck to it. All right, so this nucleotide is actually stuck on in sort of an inverted way. So we call this the 5' cap, right? This basically protects the sort of the start of the RNA molecule from being broken down, right? Because if you allowed this RNA molecule to start being broken down, you're losing the information. And that could be really bad, right? Because the information is meant to be, delivered in the form of this RNA to the ribosome, for example, to be translated, right? That would be like if you had this piece of paper, right, and you didn't somehow protect it, right, and parts of it got torn off, then you can't successfully make the product that you need to make from it. The other end of the RNA transcript, right, has a whole bunch of adenines put on the end, right? So this is what we call the poly-A tail. So you can see here at the end of the RNA transcript, you have a whole string of A's added on. And again, that's also a way to protect the end of this tail. In addition, the 5' cap and the poly-A tail serve as signals. So not surprisingly, right, there'd be certain proteins that are in your body, in the cell, in the nucleus, that would recognize these, right, attach to them and help transport the RNA out of the nucleus into the cytoplasm. Okay. Importantly, the pre-RNA, in other words, the original transcript of the RNA, may have many sequences in it that are just not used. Right? So many coding genes have stretches of DNA in them, right, that are present in the original photocopied version, right, but are no longer present in the final version that actually gets translated. So you can see here, this is a cool experiment that was done a long time ago, where scientists took a stretch of DNA that contained a gene that they knew they wanted to look at, right? And this gene, they were able to make the DNA single-stranded by kind of denaturing it, and they mixed in mRNA that is the finished edited form of RNA for this gene before it's actually turned into a protein, right? And then they mix these two together, the finished mRNA and the original gene. And what they noticed in this figure here, the red is the finished edited mRNA sequence, right? And so it's going to base pair up with the single-stranded DNA here. But notice, certain regions pair up very nicely, like this whole stretch through here, but certain regions of the DNA are looped out, right? So in other words, from this point, it just sort of skips over this whole part of the DNA that looped out here. The RNA matches. That means in the final messenger RNA, this original part of the sequence is no longer present. It's been edited out. So those parts that get edited out of the RNA are what we call introns. The parts that remain, in other words, in this cartoon here, the remaining red parts that are kept are the exons. Okay. And so this is what RNA editing is all about. Remember in the cookie analogy, I said one of the benefits of this is your friends could go ahead and edit this, right? Some of them might say, I don't want milk chocolate chips. I'm going to use something like nuts, right? Or I'm not going to use sugar. I'm going to cross this out and I'm going to put like a sugar substitute, like Splenda in there, right? They are editing the transcript, and the eventual cookies that they make, right, are a function of whatever edits they made in this transcript. And that allows for lots of variations of cookies to be made from the original starting DNA recipe, right? Likewise, from one gene that codes for a protein in our chromosomes, right, because they're first made into a primary RNA transcript, which is the perfect photocopy, that primary transcript can be edited. Right? Different parts can be eliminated and the remaining parts put together. That's how your cells make different variations of related proteins from the same gene. So that's what this illustration shows here. Here's the primary transcript, the pre-mRNA that was made by the RNA polymerase as the photocopier, making a perfect copy of the DNA sequence. Right? That RNA transcript is protected by putting on a 5' cap and a poly A tail at the end. So this piece of RNA your cell knows is a valuable piece of RNA because it represents something like the coding sequence that needs to be turned into a protein. After editing, slicing out introns and keeping exons, this would be the finished mRNA, the mature mRNA, right, or the messenger RNA that then can be translated into a protein sequence. And you can see here, starting from the same gene, depending on how you mix, match, and edit these, you get different mRNAs, right? I think we now believe that on average, each protein coding gene in the human genome, on average, can code for 3 different protein products at the end, based upon the editing of the RNA that comes off of it. Okay, so this is the last slide for today. So to sum up then, right, I want you to think about the fact that life is dependent upon all of these biological macromolecules working together, right? The 4 main types, nucleic acids like DNA and RNA, proteins, carbohydrates, and fats, right? As we said, fats and carbohydrates we get from the outside world, okay, so that we get them from the food we eat that are inside. Of course, we also get the raw ingredients for nucleotides and amino acids from the outside as well. But the important thing to keep in mind is the DNA that we have in our genes is coding very specifically for RNAs that can be little pieces of RNA that work together with the ribosome, right? Or messenger RNAs that actually contain the amino acid sequence information that gets translated by the ribosome to make finished proteins. But all of these are shapes, right? So our genes are simply discrete sets of instructions for making correctly shaped molecules that can then work together with other molecules to keep us alive, right? And those shapes are all written in the language of DNA, A's, T's, C's, and G's, right? In some cases, the rRNAs and tRNA molecules, those are the finished expression of the structures that are meant to be, you know, little sections of our genome are meant to make these shapes, right? mRNA represents an intermediate. It is sort of like the intermediate set of instructions that still need to be translated to make the final molecules intended by their shapes or their genes, right, which are the proteins. Okay. All right, let's go ahead and stop